Okay. So yeah, thanks for the invitation. Thanks for being here. And what I'm going to do, so I'm still going to tell you, I think, about prediction and immune repertoires, and I just have the wrong title slide. So let me, just a second. I'll get there because I just changed my talk because Daniel asked me to. And since Daniel and I go way back, here we go. Okay, share screen. Share screen. All right, so I mean, the purpose of the title slide is mainly just so that you see all the people who were involved in sort of working with me, and also with Thierry Morat, who's also been doing this work together. And so they're all listed, and so I'd really like to thank them. So I don't want to leave the title slide. The talk got modded at the last minute, so how it goes. But let me start with a whodunit. So remember the time like about a week ago when some of you thought you'd be at this meeting in person. Remember even further back when you would go to these amazing meetings in remote locations. Actually, case ICAM made many of them possible, and it was great, and there was usually a party. So imagine that scenario, and imagine that the next day Joe is found dead with some drops of blood next to their body that obviously don't belong to Joe. So this being scientists, they all shout genetic tests for everyone. They carry out, and they identified that the murderer or the owner of the blood twins or Terry. So normal genetic tests cannot distinguish between identical twins. Since they're identical twins, they have the same genetics. So they call Detective Asia Bear, who is familiar with modern forensic technologies, and she suggests that they should look at their immune systems. So what is your immune system? I mean, as we were talking, you're all in immunity after these last 2 years, but your immune system, your adaptive immune system, is made out of two types of cells, B cells and T cells. And these cells have receptors on the surface. And these receptors, well, they act as any receptor. So they basically have the ability to basically discriminate through this binding, discriminate what's a foreign molecule to your body, so potentially a pathogen, which then triggers an immune response and tries to get rid of it, as opposed to a protein that's natural to your body and should be there, in which case you should just leave it alone. And this is that each cell has many receptors, but they have the diversity needed to protect you against many different pathogens. You need an ensemble of different cells so that you can have different receptors, which then can recognize different pathogens. And this ensemble of cells is what we call a repertoire. And thanks to sort of relatively recent sequencing technologies that were also invented by physicists, with Steve Quake leading a major role in this, we can now just get sort out the T cells and then sequence them. So we have the sequences of these receptors, and we have insight into the natural repertoire, so the repertoire that is able to protect you in principle against the different pathogens. Now, if a given cell through its receptor recognizes a pathogen, this leads to a proliferation effect, meaning this cell multiplies and makes many copies of exactly the same cell in the case of a T cell. And so then if you look into your bloodstream, you should see many more copies of T cells that responded to something than of T cells that haven't responded to anything. And of course, cells that don't respond after some time, they have a finite lifetime, so they die. So this gives an idea where the distribution of diverse different receptors comes from. And you can also see that they come in different counts. But thanks to sequencing, we actually have access to all of that. So now if Asia takes just a thumbprint from each of she'll be able to figure out who these drops of blood that were found by Joe's body belong to by looking at repertoires. Figure out which sample comes from the same person as opposed to samples that come from different people. And she can do this just by looking essentially at the number of shared sequences between samples. So if you look at the number of shared sequences, samples that come from the same person share many more vector common than samples come from different people, including samples that come from identical twins. And
sorry, there is a problem, a sound problem. I don't know whether everybody has the same problem or whether it's only myself. So,
I have the problem too. Yeah, I was just not sure about my own internet connection.
No, it's common.
Okay, so
is that coming from the Alexandra or?
I hear a squeaking noise.
Now we hear the audio chopping a little bit from time to time.
Okay.
Maybe you can go to Saclay. Henri's connection is very good.
Okay, you want me to switch it? Okay, I'll try to switch off my video. So is the sound better now?
It depends. You have to.
We'll see.
Okay,
We'll see. Go on, and we'll see.
Otherwise, I can try to switch to my phone and see, maybe it'll be better. But okay, so so this signal that you get from the number of shared sequences being significantly basically larger if they come from the for over many decades. So meaning that if we were to test which of the twins is the killer 40 years later, we would still could figure out who the killer is. So it's a very stable signal, and of course it has to be Alex because we don't have a choice here. But the point being is that you can identify a single person out of the whole population. Don't work exactly. I have to say a little bit more about the biology of the system of what is an immune receptor. So the sort of thing to find is that you have of the order of 10 to the 9th different receptors. And if you wanted to encode it directly in your DNA, as you encode all the other proteins in your organism, it simply wouldn't fit. There's just no room in the nucleus for so many genes. The 4-5 orders of magnitude more, and that doesn't work. So what they come with is functional, where it converts randomness to generate diversity. So each receptor is made out of 3 parts called V, D, and J, and you randomly pick one V. So in your DNA, you have template for each of the genes, each of the D genes, and the J genes. One V, one D, and one J, and you get rid of the rest. So you empirically pick one example, and then you randomly insert and delete. So this gets you with the numbers of combinations we have. This gets you to about 1,000 different receptors, but we need to get to 10 to the 9th. What then happens is that randomly you delete and insert nuclei space. So essentially letters, the V and the J. And so this stochastically adding stuff in and taking stuff out gets us to 10 9th. And that if you do that, you've basically some templates which work, and they've cut them up. You've put stuff in and taken stuff out. So you can imagine that you'll end up with something that will not be a protein. It's sort of think formally be out of frame, meaning that it's into protein work. And that does happen. If we look into this data, we actually
Excuse
see
me, Alex.
some fraction of,
Alex, yes, the voice is still chopping a lot. So would you like to try your phone?
Yeah, let me just try another thing. Let me to unplug this. Is better?
There's still some choppiness there, yeah.
Okay, so let me
Right, good.
Okay. Is that better?
Yes, this is better.
Okay, great. All right. So okay, so basically you end up with these sequences that won't make a protein. And if you look in the data, you actually see them. You see these sequences that are out of frame and they won't make a protein. But the plus side of this is that these proteins were never expressed, so they were never selected upon. Because when I told you that the cells proliferate, well, there's a selection process because some of them proliferate and some of them die. And so these receptors that were never expressed could not have undergone selection. So thanks to this process, we have access to the fact that they just were generated. So we can learn something about the statistics of the generation process. Okay, so using these non-functional receptors. And of course, the reason is that they're in the same cell with a functional one. So if this happens that the non-functional one is produced, then the second chromosome, because you have 2 chromosomes, one from each parent, the second one gets rearranged, and then that one is functional. So you only get the cells where one is functional and the other one isn't. And you can learn something about the statistics of the generation process from the non-functional ones, which are sort of like a fossil record of this generation process. And so so how do we do this? Well, what do we want to learn? We want to learn the probability of generating any of these sequences. So what we could do, so we need to figure out what's the likelihood of each of these processes of picking a given V gene, picking another D gene, and so on, and having a certain number of insertions and deletions. So the simplest thing we could do is we could just figure out what's the best V gene and align it to all of these templates. But you can see that that won't work because we've come in and we've then deleted and inserted stuff. So there's many different ways in which, since you're allowed to cut and paste, basically, you can produce the same string of letters in many different ways. So there's no deterministic way of doing it, and we have to do it probabilistically. So what we do is we consider all the scenarios, by which I mean the choice of V, D, and J gene and insertions and deletions that are consistent with the sequence we observe. Then we assign weights to them, and then going over all the sequences, we calculate marginal observables, such as the probability of using a given V gene, D gene, number of deletions, insertions, and then we have a model, which is the most general factorializable model, to recalculate these weights. And we do this self-consistently until it converges. And it converges, and at the end of the day, we know the elements of the model, such as the probability of using the V gene, so on, number of deletions and insertions. And if we sum over all the scenarios that are consistent with a given sequence, weighted by these probabilities, we can calculate the probability of generating each of these sequences. So that's what we do. And this is what it looks like. This is a probability density or a histogram of the probability of generation of a given sequence. So we ask in the sample how many sequences have a probability to be generated. That's 10 to the minus 10. That's a dot here, 10 to the minus 12, 10 to the minus 15, and so on. So we get a density distribution like that. And you see that it spans 20 orders of magnitude. So some sequences are fairly easy to make over here, and some are quite hard to make. And basically, when we're looking into the twins, what we're seeing in twins are they have the same machinery for generating these random receptors, but they run different realizations of the random process. So each twin rolls their own dice and generates their own set of random receptors. And although they have the same machinery, as in fact do all of us in the world, we're going to generate different realizations. And thanks to these different realizations, we're able to identify them. But now, so this characterizes the generation process. We can similarly characterize the selection process that happens later, so which cells are more likely to survive by looking at the now functional data receptors. And then we know what's the probability of seeing any T-cell receptor in your bloodstream. And in fact, we can calculate the number of shed receptors between any number of people in our data set, which goes up to about 1,000 individuals. And we can compare the data to our model predictions. So what's the number of shed receptors? Well, it's just asking the question, what's the probability that you generated a receptor and it was selected in you? And since what happens in you is completely independent of what happens with me, by squaring this probability, we know these probabilities, we know the probability, we have basically a model expectations since we've characterized these processes, and we can do it for many people. And as you can see, the model matches the data very well. So basically, there's nothing special about you. There's nothing special about the immune receptors you produce. It's just a sort of, this is at the level of unique receptors, not taking into account the counts. It's just your one realization of a random process. So we can predict by chance how many receptors you will share with essentially anybody in the world. It's a very universal process. And you can ask, well, are we close to exhausting? So as I said, we have 10 to the 9 unique receptors. We have 10 to the 12 total in each one of us. But since we're all generating these random numbers, we essentially we have a much higher capacity. The diversity of this process, the entropy is much higher. So as humanity, as the worldwide population right now, have we exhausted the potential diversity? And so here we are today with about 10 billion people on the planet. This is what we've sampled in experiments so far, about 1,000 people. We can extrapolate that. Of course, there's a sampling problem. We don't suck all the blood out of a given person when we sample them. So the model has actually a higher number of different T-cell receptors than we're sampling, but we can extrapolate it, and the model allows us to extrapolate it. And we can see that we're nowhere close to exhausting the potential diversity of the immune system. So basically every new person, so if we were, this would plateau basically. So every new person that is born is still generating receptors that have never been seen before on Earth. So again, we're basically talking about sampling from a random process with a high diversity. And so although thanks to the fact that each person each person shares a certain number of receptors with somebody else on the planet, roughly 50 with anybody else, 50%, but 50% of your repertoire is completely specific to you, and that's enough to identify you out of the whole worldwide population. So you could ask whether the ones that we actually share correspond to anything sort of useful. For example, you could imagine that we share them because on evolutionary timescales, they've evolved to protect us against some very old pathogens. So this may be the case, but they don't protect us against any virus that is in the database. So we looked at all the viruses that are in different databases that we have, and we plotted the probability of generation of these receptors. And so this is for different viruses, receptors that are specific to these viruses, and the gray line behind is the normal probability of generation. And so basically there's nothing special about receptors that protect us against viruses, even sort of all the ones as yellow fever. But of course, maybe it could be a problem with the data, with the databases that we're not really sampling. We don't know what the old ones correspond to. Okay, if you do this kind of sequencing, you can it turns out that you can also figure out that people have COVID. So if you sample our conference participants, then we found out that both Pat and Terry had COVID, and so we ended up where we are now. But you could say, because again, you'll auto-test experts now, that, well, to figure out whether somebody has COVID, had COVID, you look at the antibodies, which are B cells and not the T cells. So, okay, so you wanted me to talk about COVID, so I'll talk about COVID. And so what we did is we found two people that went on a skiing holiday in February on 2020 and came back with COVID, so they self-isolated. And then donated blood at day 15, 30, 37, and so on till 85, post onset of symptoms. Now they had very mild symptoms, loss of smell, headache, small fever, nothing serious. They were not hospitalized. And so the T-cell repertoires were sequenced, and we then could look at how frequent a given T-cell receptor was as a function of time. This is what it looks like. Okay, so these are time-dependent trajectories of different T-cells. And you can see it's a mess. And that's mainly, as a physicist, I'll mainly be concentrating on the fact that it's a mess. So the first thing we did is just a rough scan, and we did time-dependent PCA on these we did PCA on the trajectories, and we were able to identify 3 trends. One trend where nothing happens, one trend which we expected, the green one, that there was a peak at day 15 or earlier after the onset of infection, and then it went down. So these were T cells that responded to the infection early on. And then we have this mysterious peak at day 37, a secondary peak, which we were not expecting and we still don't understand, but since then we've seen it in other people. Okay, but we're getting this from data that looks like that. So as you see, it's a mess. And so we wanted to be a little bit more certain that what we're doing makes sense. So we figured we need to actually calibrate the noise. So let me give you an example of what the noise looks like. These are 2 samples, so there's 2 eggs everywhere here, which means that at every time point, we got 2 samples from the same persons precisely to calibrate the noise. So this is Terry on day 15, and this is the number of count for each T cell in the first sample and the second one. So this should basically be a diagonal, because we're looking at the same person at the same time point and asking what the counts are. But so this is, if you want, this is the immunological definition of a diagonal, where you have in one sample 1 cell and in the other sample of the order of 100 cells. So there's a lot of noise here. And so there's noise which comes from the experiment, there's sampling noise, of course, but there's also biological noise, because what's going on here is the reading of signals, the sequencing mRNAs, and mRNAs have intermittent or bursty expression. So sometimes they're produced in bursts and then nothing happens. Well, they make proteins, they're dead, they die off, and then there's another burst coming. So we also know that the distribution of the underlying frequencies of these receptors, of these T-cell receptors, is a power law. It scans over, well, it scans whatever your sampling is, but basically sampling out up to 10 to the 5 different cells, it's a very well-defined power law. So we know that the underlying distribution is a power law, and we know the reads are what we actually see in experiments. And so what we do is we build a noise model where we translate the power law into reads by taking into account the fact that we're sampling random cells, and that then there's a sort of bursty or long-tailed noise coming from this mRNA expression, which we model by a negative binomial. And so putting all this together, at the end of the day, we get the probability of seeing a given cell at a given size in the data. And we learn this model based on the same time replicates, and we use it as input for probabilistic inference of response. So then between time points, we have an expansion model where we say that the frequency at a later time point is related to the frequency at an earlier time point for an expansion coefficient. And we say that some fraction of the cells are expanding and some are not expanding, as we saw in this initial analysis. And then we use the posterior to identify expanded clones. And this is what it looks like. We reproduce the trends. We reproduce this contracting trend from day 15 with a peak at day 15 or slightly earlier that we saw before. But now I'm also plotting data for these people that we had of the repertoires of a year and 2 years before COVID was ever detected on our planet. So we know this is actually a peak for these cells. And then we also reproduce the second peak that we saw before. And we see a large expansion with responding clone. Well, this doesn't really matter so much. So let's take a look at this in a little bit more detail. So we can also separate these cells into what are called memory cells. So memory cells are the ones that after you get an infection, some fraction of your cells are set aside, given a special flag which says, I've seen an infection, so I've been to war. And the next time you see a similar infection, they respond more quickly than sort of basic cells that haven't done anything. So we can look into these cells, the memory ones, and we see stable memory of cells that we identified are linked to this infection after the infection. So we see stable post-infection memory. But more interestingly, we have this data from before COVID. And so from 2017, 2018, and we still see cells that are identified as responding to COVID before COVID ever hit the planet. So these two people who had mild symptoms seem to have basically immunity before they got infected. And maybe that's why they got mild symptoms. We don't know about that. I'll get back to that. I don't have to talk about this since this is a last minute talk. Basically, they get stable memory. Okay, but what I'm telling you so far is basically a statistical analysis to identify something that's responding. And a fair question is, how do you know it's responding to COVID? So what our collaborators did well, actually, it turns out that, well, what we did, but it turns out that Pat, one of the people in our study, actually had participated in another study 20 days after they had an onset of symptoms. And this is this study is a sort of fishing expedition. So what they do is they take a molecular fishing rod and they put a COVID protein, or specifically an epitope, on the end of this fishing rod, and they stick it into Pat's blood. And then they pull out all the T cells that bind to the COVID protein. But it's just one protein. So in your body, you have many COVID proteins, but here they put in one specific one. So here, if you care to know what it is, this is what it is. And so they pulled out a series of T cells. And then we took these T cells they pulled out at day 20 and asked whether we see them in our data sets at different time points. And so the dots are the ones we see, and we reproduce this general trend. So this contracting trend that we were able to identify just from data, and the green ones are the ones that we identified by our purely computational methods. So we're basically managing to get most of the high frequency clonotypes computationally without any need for experiments. And we have a patient specific way for identifying COVID responding clonotypes. Okay. So, so we can be pretty certain that most of what we are identifying as a strong response is linked to COVID, but we can do another test where we take the cells that we identify and mix them with results of a large scale fishing experiment. So now they take many COVID proteins and they put it in the blood of many people, and they pull out the T cells that respond to them. And so we have now T cells that, again, respond to COVID. We also know what protein they respond to. We mix these cells with the cells we identify as, like, computationally. We put them together as the ones that respond to COVID, and we build similarity networks. So we basically put links between T cells that differ by at most one amino acid. And this way we identify clusters for which, so we clustered this data this way, and we identify 32 clusters which correspond now to 32 different COVID or SARS-CoV-2 epitopes. So this is what some of them look like. And let's just focus on this pink one here, which is Pat's what's called immunodominant epitope, which means that 21% of all of Pat's response is geared at identifying, at binding this one COVID protein. This is what the Tcell motif looks like. And what's interesting, well, the reason I'm telling you about this, because you're like, well, why is she showing us all these letters and so on? So this is the COVID protein that corresponds to this cluster. But T cells that were specific to it were already there in Pat's immune system a year before the infection. And it turns out that if you make a one amino acid mutation in this protein, you end up with a common cold coronavirus protein. So our suspicion is that Pat got the cold, developed T cells against it before COVID, and that helped them have a mild infection later on. Okay, so I'm gonna skip this. And going back, so this is the sort of original thing that I wanted to say about COVID before I added the previous stuff. So going back to the fact that we can predict how many T cell sequences are shared between how many people. This is sort of a replotting of what I showed you before. We can also do it for B cells, but it turns out that if we do it, so this is B cells in healthy people. So B cells is another arm of the immune system. Basically, for the purposes of this talk, you can think about them as T cells. but if we look at B cells in people who had COVID and ask how many sequences they share, this is our theoretical prediction. And this is how many, the dots are how, the crosses are how many are actually shared in real people. So people who had COVID share many more B cells than predicted by chance. And this is something we know, that COVID focuses your repertoire. So in a way, it gets rid of diversity in it. And even though you're a very different person, you respond very similarly to it. Okay, so that's all I had to say about COVID. And so let me get back to the original topic of my talk, which is prediction. So while we've been talking about the immune system, viruses, of course, evolve. Again, something that, you know, I don't need to convince you about. This is actually a map. This is taken from Trevor Bedford, who by now is probably a household name also. This is the transition matrix for the flu at the point when we still cared about the flu. And these are the major airline routes. So you can see easily the flu travels by plane. And that's a known fact. And it travels a lot around the world. And the question we were interested in asking is, can we estimate, well, basically, can our immune system predict future strains of the flu? So we know that as scientists, having the worldwide data, we can predict on the timescale of a few months, some future strains of the flu. But can our immune systems actually do that too? Meaning, are they trying to read temporal cues from the past and adapt in such a way as to best prepare ourselves for the future? And so what is prediction? Prediction is essentially estimating frequency. We're trying to estimate what will be the most dominant strain in the future. And so these are the power laws that I mentioned before in human T cells. This is in zebrafish data. We see it everywhere. So these are the power laws in say, T cell frequencies. So can we readapt our composition of the immune repertoire in such a way that these ones that are quite frequent are best suited against the future strains that will come? And estimating frequencies estimating frequencies is basically a problem that all of these kinds of diverse computing systems, such as the microbiome or any sort of ecological problems, also halt. And the problem goes back to the Middle Ages, when kings wanted to know how many different kinds of animals they had in the forest. And it's not a simple problem because of sampling, and the fact that we're trying to estimate something from a dynamical stochastic process. So, okay, so basically, what are the elements that we need for prediction? The first thing we need for prediction is we need the process that we're trying to predict to actually be correlated in time. If there are no correlations, we cannot predict anything. So what we're basically asking here is, if we have such a composition right now of the immune repertoire, how should it change? So which of these cells should become more frequent to be best adapted against the future viruses that are out there, assuming that they're sort of okay against current viruses. And for that, we need the viruses to be correlated in time, which they are because they evolve through sort of evolutionary dynamics. And we can talk about how COVID fits into that in the discussion. Now, the other thing, so you need correlations to be able to predict things. The other thing you need to be able to remember things. And so, as I already mentioned, the immune system can remember things. It has a memory repertoire where cells are set aside if they've seen an infection. And so the first question is having this memory repertoire useful? Is remembering stuff useful? And if yes, how should you update the memory repertoire? Should you update it proportionally? That meaning every time you see the same pathogen, you keep the same number of cells, like 2 here? Or should you have a modulated response, meaning that the next time you see something, you pay a little bit more, a little bit less attention to it than the first time you see it? Okay, so how are we going to formulate the problem? We're going to say that there's some distribution of pathogens out there that evolve to according to some stochastic dynamics. You can think about Wright-Fisher dynamics if you want. And we, on the other sort of team, we have a receptor distribution whose name is to sort of recognize pathogens. So what the name of the game is to minimize the expected cost of an infection. So to minimize the cost between the repertoire distribution and the pathogen distribution. So what we're going to try to find is what is the optimal repertoire distribution that minimizes this cost. Now, the only problem is we don't know the pathogen distribution, right? So we don't know what we're trying to fight. So what we're going to do is we're going to introduce a belief in the sort of formal mathematical sense of the word. So a meta distribution over the distribution of pathogen frequencies. And we're going to integrate over our belief of what these pathogen distributions should be. So what we're basically, and we're going to update the repertoire according to this belief. So basically what we're saying is we're assuming that our organisms, that our repertoire holds a belief about the state of the pathogen distribution, which it dynamically forecasts in time according to the same dynamics that it's learned on evolutionary timescales. So it updates it, but every once in a while it actually interacts with the real pathogen environment, and that means it samples it. That means that also it collapses the belief in that certain point. So it no longer needs a distribution about whether that pathogen exists or not, because it's actually made a measurement and it knows. So it collapses the distribution and it continues to forecast or propagate the rest of the distribution. So we're saying that prediction is basically a combination of having a prior that you propagate and verification of this prior locally in terms of encounters. And we can do that thanks to the memory repertoire, which allows us to remember what we've seen and keep track of these encounters. So the missing piece, of course, is what is this cost? And so there's 2 elements of the cost. So now I'm going to assume that and consider this problem in an abstract space that I'm going to call recognition space, and or shape space if you're familiar with the concept. But basically it's a phenomenological space that if a receptor and a pathogen are close to each other in this space, they recognize each other. And I'm going to measure the time in the number of encounters. Actually, I don't see my slides. Okay, good. Sorry. Yeah, and right. So if a receptor and a pathogen are close, then they recognize each other. So for example, this blue receptor will recognize the blue pathogens, but there's a concept known as cross-reactivity, meaning that the pathogens that are here between the blue and the red can be recognized by any of these, either by the blue or the red receptor, meaning that there's not a one-to-one mapping, there's a degeneracy. So you can think about each receptor being able to recognize some cloud of different pathogens. And this is what I mean by this recognition space, that basically the closer they are, the stronger they will recognize it. So you can think about these as little Gaussians. So then the probability of an immune response from an encounter with a given pathogen, you have to sum over all the receptors that are able to recognize this specific pathogen A. Okay, And then we just encode the fact that the harm caused by an antigen generally increases with time, meaning you're trying to kill the antigen or pathogen, and you're not succeeding, and then you're not getting any better. The cost to your organism, the cost to your tissues increases. And so we encode this in a way that we say we have Poisson distributed recognition events, and we integrate over the effective cost of an infection, which we consider different functions, but generally it's a non-decreasing function of time. And we integrate over time, and we can correct for having different virulence factors. Okay, and so that's the cost from one pathogen, and then we just have to sum over all the pathogens we encounter and integrate over our belief of what the distribution is, and we minimize the cost. So let me go back. So, okay, so we do this numerically in two-dimensional space, and we look at the relative cost decrease compared to not having a memory, not being able to remember and foresee things as a function of how many pathogens are sampled. And we see that it's a huge decrease for if pathogens are sparsely distributed in this recognition space. So basically memory helps in sparse environments because it's easier to detect these few sparse pathogens, and you don't need to fill in the details. Of course, similar pathogens are easier and reduce the cost more than pathogens that are more different. So the faster they evolve, the harder they are to recognize, not surprising. But what is surprising is that if we can take all these parameters that we've been varying, we can collapse all of these curves onto one curve. And the only thing that matters is there's two parameters that matter: sampling and sparseness. There's a combination of these parameters, but basically those are the only two parameters that matters, which is reminiscent of compressive sensing. So the fact that it's easy to reproduce images from sort of randomly, well, badly sampled data, noisy data, if the more we sample, of course, but also if the data is sparse. And so that's something similar that's going on here. It's easier for the immune system to recognize things if in sparse environments. And basically what's going on, you're quickly learning the global features of the distribution, and that's what you see in people as a function as a function of age. Basically, this is the memory fraction, how it goes up. So you learn the sparse features early on, and then as you get older, you're basically filling on the holes, and it's getting harder and harder. And to answer the question,
would you like to summarize in a few minutes? So we
I'm finishing. I've just answered this question, and yeah. So basically, so to answer the question of was memory useful, yes. And every time you see and the same pathogen again, you should respond to it less, which gives you the strong negative feedback in the system, which we see in the data. Okay, so I'll skip all this. Okay, maybe I'll just say this. So what do these optimal repertoires look like? Well, if this is the antigen distribution in red in blue in 1D, the optimal repertoire is peaked. So you get these Dirac combs, and you get the same speckle-like pattern in 2D. And so then there's a question: if two individuals see the same environment, should they have the same optimal repertoire, or should they have different optimal repertoire? So say you see the blue and I see the yellow. And the answer is they can have very different optimal repertoires, because the exact positioning of the receptor doesn't matter. So this is what I draw with this cartoon here, where I draw the lines to put the receptors in different places, but it doesn't matter exactly where they are. In fact, the similarity between these two optimal repertoires goes down very quickly as a function of how we see the environment, although the antigen distribution we're seeing is very similar, and the repertoire, basically we both have perfect protection. So to bring back this to the data, although now I'm talking about phenotypic space, I showed you before, this is a sort of a different rendition of how many receptors we expect two people to share. Well, we can predict it, but these numbers are fairly low. And that's what we expect from this theory, that again, due to these random realizations that don't have to be perfect, but just have to cover space, we can have very different solutions to the same problem. So that's it. I just sort of wanted to tell you about how, although the system is very diverse and very stochastic, we can still find statistical rules in it and make stochastic predictions. So I'll stop here in the interest of time.
Well, thank you, Alex, for a beautiful talk. The floor is open for questions now. People can either put something in the chat, or they can raise their hand or unmute themselves and ask questions. Jose?
Very nice talk. Really enjoy it. I'm just curious about your entire prediction thing, how we do the idea. Having memories, it's to some certain point you have too much memory to become a problem. That's basically you have to saturate. That was hard for me to get from the equations, basically. Have no memory is a problem, but have too much of them is also, you have too much checking to do first, right?
Yeah. No, so let me see if I can pull up a slide. Maybe I can't pull up a slide on this. But yeah, I mean, one of our, you're absolutely right. And so it's very important to forget. So, and you know, so you do need, so you do forget. And of course you should, the faster your environment changes, the faster you should forget. But if you don't forget, you're completely right. You have a big.
But how our system does that? For some diseases you have immunity for your life, and some others you don't. How do your
that's a
system
great question, and you talk to immunologists and they don't know. So the basic version of this question is, why for some things do we just get a shot as kids and we get a series of shots and we're fine? And then, you know, like for tetanus, we need to get revaccinated every 7 years. Why is it every 7 years? It's a completely empirical fact. Okay. And it's like, you know, now you're experiencing the same thing with COVID, right? I mean, initially they said wait 6 months. Now it's okay. 3 months is okay. You know, it's black magic. We don't actually know. Okay, that's a bit different because they evolve. But even for things that don't evolve, what is the time scale for, you know, for decay of memory? We don't know. And you know, you can understand why it may be hard to measure it. You could measure titers, but yeah, there's no really quantitative data on this.
Do you have any clue why, or is that just something that basically?
So why, I'm guessing, is a sociological reason that, you know, they can figure out through basically looking at people that, you know, you need your tetanus shot every decade.
No, that's empirical. That I can understand that.
So empirical works fine. And then why they don't push to, I don't think it's a hard, I mean, I would know how to do this experiment, right? You just get titers for a specific strain. Titers are basically how, you know, how strongly you're responding. There's now easy way of getting titers. And no, I mean, now people are doing it for things that mutate. But that's a different question. So one part of the answer is we know memory cells die. We know they die relatively quickly. We know there's two concepts. One is homeostasis, so the size of the memory pool is fairly constant, and we know that initially you're filling a void. So when you get infected with something as a kid, that will probably last for longer because you can just keep more memory. As you get older, you have to you have to push and shove to keep those memory cells. And that we see in various data. But again, what would be great is really quantitative measurements about how this decays. I don't know about that data.
And that's just great, but the decay is not uniform from disease to disease, right? You said that's one thing.
That's clear, yeah.
I have a question about something you mentioned about our immune system trying to extrapolate the evolution of the flu virus. So when we have a seasonal, is it because, first of all, flu is a seasonal thing that keeps coming back every year? So somehow our immune system is thinking about what would be the new strains of flu. And how successful is it in doing the extrapolation?
Yeah. So first of all, this is theory, right? We made the assumption what would happen if our immune system was trying to predict it, and then we can make predictions in that framework that agree with what we see in various data, right? But right. I think it's still, it's not definitive proof that our immune system is actually successfully predicting or trying to predict. We know it evolves, we know it has dynamics, and so far this dynamics is consistent with a system that is trying to predict, okay, the way it updates it. How successful is it? Well, you don't get the flu, the real flu. We all say we get the flu, but what we really get is a cold. You don't get the real flu that often. I mean, if you get the real flu, you're in bed for a week and you feel really bad. Now, so probably you are able to, your immune system. So again, if I were to interpret this, there's a correlation time scale with this year's flu being similar to last year's flu and so on. And if you're within the correlation time scale of still having protection, you're fine. And then, something new comes around. And in fact, then it gets more complicated because there's different strains. And but that's really the reason why if a flu comes from a different reservoir, that's why that's when we have an epidemic, right? That's what we had with the bird flu. That's what we had with MERS, well, which isn't the flu, but okay. But you're saying what hopefully you're understanding what I'm saying. So there does seem to be signal that if there is continuity in the flu strains and you're protected against it, you're fine.
Yes, has a question.
Yeah, so this is an ignorant question from a theoretical physicist who's very far from what you work on, but the information that gives us the genetic information you're referring to is stored on the DNA of the lymph cells. Is that what you were saying to us? So they actually modify their own DNA, yes, to put this information in. Is it on a specific chromosome or is it in all the chromosomes?
No, so it's on a specific chromosome. So basically, you learn at some point in your life from having to listen to all these talks that we all have the same DNA. sorry, so that's not true. That all the cells in our body have the same DNA,
Yes.
right? So that's true, except for immune cells. So these lymphocytes, so lymphocytes are T cells and B cells.
Yes.
They actually have edited DNA. They really cut the DNA. It's on a specific chromosome. It's in a specific place, and they make little loops physically and then cut out the stuff in the middle. So you have a lot of edited DNA in your body, and you keep on editing it for your whole life.
And having edited it, they reproduce that DNA, and that keeps the memory going. I see.
So that's the thing. That's when you edit it during the generation process that actually happens in your thymus. Then they let out into the periphery, so into your bloodstream. And then when they proliferate, when they basically divide, make copies of themselves, then they only have this DNA to pass on to the cell offspring. So they're gonna have the same DNA. That's the story for T cells. And then even if they become a memory cell, they'll still have the same DNA. In the meantime, you're generating new ones with different DNA. Now, that's the story for T cells. For B cells, it's the same story, only when they proliferate, they can actually additionally introduce point mutations. So then they build these evolutionary lineages that they start off with the same largely edited DNA, but then they can even further modify it. So they form families of cells that have similar DNA.
I see. So you have a whole society of cells with different DNA distributions.
Yeah.
I see. That's very interesting. Where's a good place for a physicist to read about this? Where's a good reference?
We wrote this review with Grégoire Tanbonnet and Thierry Morat in Physics Reports that I definitely do not recommend as a one-time read. It's about 100 pages long. So go to the specific chapter you're interested in. There's a chapter on repertoires, and hopefully starts around page 40 or 50. So
good.
But you can go straight there. The chapters are written in a way that they should be standalone.
Fantastic. Okay, thank you very much.
We have a question from Andre.
Alexander, thanks for the fascinating talk. So when it comes to to decay or maybe the death of the cells, right? What is the half-life, so to speak, of the T cells? In other words, from your response to Jose's question, I'm trying to figure out what determines the residual immunity to the disease. Is it the memory installed in a memory cell? Is it whether or not the part of the DNA in the lymphocytes that generates a specific protein that can bind to a receptor, was it that has been lost? Or was it not T cells at all, was it the B cells still exists in your blood?
Okay. So I think that the short answer is everything. So of course, also T cells and B cells, I mean, they're sort of like independent arms, but they talk together, they work together. So there's communication. We now know there's also quorum sensing. So there's a population response. There's, things are complicated. But okay, let's start with the lifetime of a T cell, because this is something that we actually did. So I can tell you in more detail if you're interested, but the answer is 40 years. Okay, so what that means is that you yourself, looking at you probably have T cells that were generated before you were born.
Wow.
Okay, so T cells. So remember, I told Jose also that you start off with an empty bag. Now, what happens is in embryonic life, you start producing T cells, but you produce T cells as an embryo that don't have these random insertions. They just do the combinatorics and the deletions, but they don't do the insertions. And working with our Russian collaborators, we were actually able to then track these zero insertions T cells and their overrepresentation. And from this, we were able to determine the half-life, basically. And so it's 37 years, so it's about 40 years. So that's the answer for T cells.
In that case, the T cell generated, let's say, in response to tetanus shot should still be there 7 years later,
Yeah.
and even longer. So it's not the T cell that's the limiting factor?
Well, so a lot of vaccines are B cell based. So that's one answer. Okay. But remember again what I said about the empty bag. This is coming from a time when, when they were produced and they could just expand. They also expand a little non-specifically. So by the time you get, say, a tetanus shot at the age of 30, well, they have to fight with each other. And I mean, honestly, the short answer is we don't understand. That's why it's an empirical fact. You get it every 7 years. Of course, nobody does, right? But you should get it every 7 years. And why it's 7 years for tetanus, but it's once in a lifetime for polio. I mean, you get a series of shots, but you never get polio reboosters, right? I mean, I've never heard a convincing or even an attempt at an explanation. So, okay, so there's a lot of, when I was talking about the mess and I was showing you the data from the COVID as a function of time, that's really what it looks like. And so one thing we're trying to do is we're trying to characterize the dynamics after an infection, after some strong stimulation. The other thing we're now trying to do is actually understand the dynamics of nothing happens, because nothing happens is not nothing happens. That's what you see in the background there of all these clonotypes going up and down. And we have a characteristic time scale for that too. And we have a noise factor, and I don't remember it off the top of my head right now, although I was looking at data earlier today. But it's definitely not, nothing happens is a lot happens. And it changes with age. So, and I don't actually remember which way it goes. But yeah, there's a strong function of age about how this dynamic happens, because as you get older, you get wiser. So your memory pool gets a little bit bigger, but very, very much filled up. But because of that, you have a harder time dealing with something completely new. Okay, there's also anecdotally, there's this thing called CMV, which is a herpes-like virus that the fraction of people in the world that have it is proportional to their age. By the time you're 80, I think 80% of people have it. Generally, a lot of people have it. But also by the time of you're 80, the majority of your T cells are dealing with just controlling this virus. It doesn't kill you. But your immune system gets obsessed about controlling it. And the majority of your T cells just go to control it and keep it under control. And then you die from the fact that you don't have T cells left to do anything useful. So, the short answer is these, we don't know. This is what I try to do in my research, to come back in a few years and maybe give you a better picture but the dynamics is different for different clones. There's basically stationary dynamics. The response dynamics is much stronger. The fraction of cells that respond to an infection, even a long-term infection as HIV, is a small part of the repertoire. How is that controlled? We don't know. So, sorry.
No, this is great. Thanks.
Well, thank you, Alex, for a great talk.